You know that sudden jolt of panic you get right before you lean in for a first date, or maybe it hits you just seconds before you have to whisper something to a colleague in a completely silent, crowded meeting room,
Oh, absolutely.
right? It's that universal, deeply human fear. Like, what if my breath is terrible right now?
It's a profound social anxiety. And, the funny thing is, it's completely justified statistically.
Wait, really?
Yeah. If you look at the primary reasons people actually go out of their way to sit in a dentist's chair seeking treatment for oral malodor, which is the clinical term for bad breath,
Hmm.
is right near the very top of the list.
Wow.
It's sitting right there behind treating active cavities and severe periodontal disease. I mean, it is a massive driver of dental visits.
Right. But usually what happens when we feel that panic? We don't go to the dentist immediately.
No, of course not.
We run to the pharmacy, we stand in the oral care aisle, and just stare blankly at this overwhelming wall of brightly colored liquids.
All the neon blue and green bottles.
Exactly. Mint explosion, wintergreen, Arctic freeze. I mean, it is almost entirely marketing. You're just buying a temporary flavor.
Yeah, totally.
But instead of just grabbing the strongest, most eye-watering mint bottle today, we are going to look at what actually works biologically. So for this deep dive, we have our hands on a rigorously designed 2018 clinical trial. It was published in Clinical and Experimental Dental Research.
That's a fantastic paper.
It really is. And we aren't just talking about masking bad breath here. We are unpacking the exact microscopic mechanics of how it is created and examining a very specific chemical, 0.1% chlorine dioxide, or
Oh.
ClO2.
Right.
We want to see if it is, in fact, the ultimate assassin for halitosis.
And to really understand why a specific chemical might be an assassin, you first have to completely deconstruct the target.
Okay, lay it out for us.
Well, for decades, the mainstream understanding of bad breath was incredibly reductive. The common assumption, even among some health professionals back in the day, was that bad breath was just the smell of leftover food particles literally rotting between your teeth.
Like an old piece of garlic just hanging out.
Exactly, but it isn't.
Yeah.
The biological reality is so much more complex.
Wait, so the source is much weirder than just old garlic or a piece of spinach caught in a molar. Because reading through this research, the smell is actually the end result of a highly organized bacterial assembly line.
Yes.
You have billions of bacteria living in your mouth, and like any living organism, they are constantly eating, metabolizing, and excreting waste. And in the mouth, that bacterial waste takes the form of volatile sulfur compounds, or VSCs.
VSCs, yeah. That's the real culprit.
Right. And the study zeroes in on two major offenders: hydrogen sulfide, which smells notoriously like rotten eggs,
Ah, the worst.
and methyl mercaptan, which smells like rotting cabbage or, honestly, feces. Those invisible sulfur gases are what actually hit the nose of the person standing next to you.
Precisely. And for a very long time, the scientific community spent most of its energy pointing the finger at one specific class of bacteria as the sole culprits for this gas, gram-negative bacteria.
Negative. Okay.
Yeah, these include strains with somewhat intimidating names, things like Fusobacterium nucleatum, Treponema denticola, and Tenorella forsythia.
Right.
Historically, dentists and researchers viewed these gram-negative strains as the primary villains of the mouth because they are the ones physically producing those foul-smelling, volatile sulfur compounds.
But that's where the 2018 study completely flips the script, right?
Exactly.
It highlights that those gram-negative bacteria, they do not work alone. There's an entirely different class of bacteria living right alongside them, the gram-positive bacteria, specifically a strain called Solobacterium morae.
Yes, Solobacterium morae.
And on their own, gram-positive bacteria are actually pretty harmless when it comes to odor. They don't produce those nasty sulfur gases.
Yeah.
But here is the biological catch. They possess a highly specific enzyme called beta-galactosidase.
Right.
And this enzyme acts like a microscopic meat cleaver. It allows them to deglycosylate, or basically chop up the complex proteins floating around in your saliva.
Exactly. And we have to explain why that chopping process is so critical. You see, the proteins in your saliva are huge, complex molecules.
Okay.
The gram-negative bacteria, the ones that actually produce the bad smell, they cannot eat those large proteins whole. They physically can't digest them.
Oh, they're too big.
Right. They need those proteins broken down into smaller individual amino acids. Specifically, they're looking for amino acids like cysteine and methionine, which, importantly, happen to be incredibly rich in sulfur.
So if we look at this like a twisted microscopic culinary assembly line,
I love this analogy, yeah.
The gram-positive bacteria, the Solobacterium morae, are your prep cooks.
Right?
They are diligently taking the raw ingredients, the saliva proteins, and using their enzyme cleavers to chop them up into bite-sized, sulfur-rich amino acids.
Yes.
Then they slide those prep ingredients over to the gram-negative bacteria. Those guys are the chefs, taking those amino acids, cooking them down, and pumping out the foul-smelling volatile sulfur compounds into your mouth.
That culinary analogy highlights exactly why the historical approach to bad breath was flawed.
How so?
Well, for years, people argued over which specific lone wolf bacterium was causing halitosis, just looking for the one bad guy.
Right, the one chef.
Exactly. But this research proves it is a highly collaborative, interconnected microbiome ecosystem. The prep cooks and the chefs absolutely rely on each other. So, logically, if you want to eliminate the smell, you don't necessarily have to kill every single bacterium in the mouth.
Sure.
You just have to disrupt their specific synergy. You have to break the assembly line.
Which sounds great in a laboratory petri dish,
Yeah.
right? But how do scientists actually prove a specific mouthwash can break that assembly line in a real human mouth?
It's tricky.
Very. The methodology of this study, which took place at the University of Medicine and Pharmacy in Ho Chi Minh City, is fascinating because of who they chose to test. They didn't just grab random healthy students off the campus who happened to eat a garlicky lunch. They specifically screened for 39 students who had genuine chronic halitosis as their chief medical complaint. These are people whose baseline state is severe bad breath.
And that distinction is vital for the integrity of the data. If you're testing a cure for a chronic condition, you need subjects who actually suffer from that condition constantly.
Right.
And the framework they use to test them is what we call a double-blind crossover study.
Break that down for us.
Sure. They took these 39 students and randomly split them into two groups. Group A used the 0.1% chlorine dioxide mouthwash twice a day for 2 weeks. Group B used a control rinse.
Just a placebo.
Yeah, it was just a 0.9% sodium chloride saline solution, but it was flavored to taste identical to the active mouthwash.
Okay, so double-blind means?
Double-blind means neither the researchers handing out the bottles nor the students using them knew who was getting the real chemical and who was getting salt water.
And what I found brilliant is that they didn't just stop after 2 weeks, compare the groups, and publish the paper.
They didn't.
They implemented a 4-week washout period. They told everyone in the study to stop using the mouthwashes entirely for a full month.
Which is such a great design.
Right. The goal there is to let their oral microbiomes naturally return to their baseline state of chronic bad breath. And once they were back to square one, they flipped the script. The groups swapped treatments.
Yep.
Whoever had the real chlorine dioxide the first time got the fake saline the second time, and vice versa, for another 2 weeks.
And that crossover design is really the gold standard here. In a traditional study, you might have genetic or lifestyle differences between Group A and Group B that completely skew the results.
Like maybe Group A naturally drinks more water.
Exactly. Or Group B eats more sugar. But in a crossover design, every single subject eventually serves as their own control. You are comparing a student's breath on the active chemical directly against that exact same student's breath on the placebo. It eliminates all those external variables.
And speaking of variables, we have to talk about how they actually measured the bad breath. Because they used 2 entirely different methods.
Yes, they did.
First, they went high-tech. They used an advanced machine called an oral chroma.
The oral chroma.
Right. This device literally draws a sample of the ambient air from inside the subject's mouth and uses gas chromatography to measure the exact nanograms of those specific volatile sulfur compounds floating around. It gives you hard, undeniable molecular data.
It does. It's incredibly precise. But, as advanced as the oral chroma is, they didn't stop there. They also utilized organoleptic measurements, which is the clinical term for a highly trained human judge using their nose to smell the student's breath.
And okay, I have to jump in here because reading the methodology for this part sounded almost like a psychological experiment.
It's intense.
The human subjects had to sit in a room with their lips tightly sealed for 3 full minutes, letting the gases build up in their mouths. Then they had to exhale through a paper tube directly toward the face of this highly trained judge. The judge then scores the smell on a rigid scale from 0, meaning no odor, all the way to 5, which is defined as extremely strong malodor close to saturation.
It's quite a job.
And I just have to ask, wait, We just established that they have highly sensitive machines that can measure exact nanograms of gas.
Right.
Why on earth are scientists in a modern 2018 clinical trial still forcing human judges to smell chronic bad breath through a paper tube?
I know, I know. It sounds incredibly medieval, I agree. But it is fundamentally necessary.
How so?
Well, you have to remember what we established at the beginning. Halitosis is largely a social disease. It is about how your breath is perceived by the people around you in the real world.
Oh, I see.
A machine can measure the precise chemical concentration of a gas, but grounding the data in human perception, the organoleptic test ensures that a chemical reduction on a spreadsheet actually translates to a noticeable difference for a person standing next to you.
Right.
Think about it. If the oral chroma machine says the sulfur gas has been reduced by 90%, but the human judge at the end of the tube still gags and smells something awful, the mouthwash hasn't actually solved the patient's primary problem. The human nose is the ultimate arbiter of success.
It's funny, it sounds archaic, but you're totally right. If the machine says my breath is chemically fine, but my date still leans away from me at dinner, the machine's data is totally useless to me.
Exactly.
The social stakes are the real stakes.
Yeah.
Okay, so we have a bulletproof study design and a ruthless measurement system. What is this chemical actually doing in the mouth? Because 0.1% chlorine dioxide operates very differently from your standard alcohol-based mouthwash.
It operates on a completely different biological level. Chlorine dioxide, or ClO2, is a highly soluble, water-stable free radical.
Okay, water stable.
Yeah, and that water solubility is its first major advantage. The bacteria in your mouth don't just float around in the open, they actively hide. They build sticky, protective structural shelters called biofilms, which we commonly call plaque.
Right, plaque.
A lot of standard antimicrobial agents in commercial mouthwashes just sit on top of that plaque. They can't get inside. But because ClO2 is so highly water-soluble, it easily penetrates deep into those sticky biofilms, reaching the exact microscopic pockets where the bacteria are actually hiding.
And once it gets inside that biofilm, it doesn't just blindly everything in its path. It is incredibly strategic.
Very targeted.
Based on the research, it attacks the bad breath assembly line on 3 distinct fronts. First, there is nutrient starvation.
Yes.
We talked about those prep-cooked bacteria chopping up proteins into sulfur-rich amino acids, specifically cysteine and methionine, to feed the chefs.
Right.
Well, ClO2 aggressively oxidizes those specific amino acids. It completely alters their structure. So it literally destroys the exact nutrients the odor-causing bacteria need to survive.
Yeah.
They essentially starve.
And then the second front is direct chemical neutralization. When ClO2 is introduced to the mouth, it releases free chloride ions into the oral environment. These ions are highly reactive. They physically interact with any volatile sulfur compounds that the bacteria have already managed to produce.
So the stuff that's already smelly.
Exactly. Through an oxidation process, the chloride ions force those smelly sulfur gases to convert into completely odorless chloride forms.
Wow.
It doesn't just cover the smell up, it fundamentally changes the molecular structure of the odor itself.
And then there's the third front, which I think is the most fascinating. Environmental alteration.
Oh, definitely.
ClO2 introduces a highly oxygen-rich environment into the mouth. And those gram-negative bacteria, the chefs actually making the VSCs, they are mostly anaerobic.
Right. They absolutely hate oxygen.
In fact, oxygen is toxic to them because they lack the enzymes to defend against oxygen-free radicals. So if you think about that standard bottle of alcohol-based blue mouthwash sitting on your bathroom sink right now, that is essentially just throwing a heavy mint perfume over a trash can.
Yeah, that's a good way to put it.
But using chlorine dioxide is like sneaking into a restaurant, stealing all the bacteria's raw food supply so they can't cook, tearing down the walls of their kitchen to flood it with toxic outside air, and simultaneously chemically deleting the smell of the food that's already sitting in the dining room.
That restaurant analogy perfectly captures the multi-targeted mechanism. And, it highlights a really elegant approach to medical treatments in general.
How so?
The beauty here is targeting the environment rather than just trying to indiscriminately poison the bacteria. By removing the specific amino acid precursors and flooding the area with oxygen, ClO2 essentially stops the production of halitosis before it even has a chance to begin. It breaks the supply chain completely.
So the theory is brilliant. The biological mechanism makes total sense. But a theory in a lab is one thing. I want to know what actually happened to the breath of those 39 students in Ho Chi Minh City when they used it. Let's look at the actual numbers.
The numbers were genuinely dramatic, and the shift happened incredibly fast. Let's look at that human sniff test first, the organoleptic score. Before the study started, the baseline score for the experimental group was an average of 2.67 out of 5.
Which is bad.
Yeah, according to their scale, that is a very clearly noticeable offensive malodor. But just 12 hours after taking their first rinse with the 0.1% ClO2, that score plummeted to 1.64.
In 12 hours.
Yes, and after 2 weeks of continued use, it dropped all the way down to 0.95, which the human judges categorized as barely noticeable. Meanwhile, the control group using the saline rinse saw absolutely no significant change in their scores.
And the high-tech machines completely backed up the human noses, right?
They did.
The levels of hydrogen sulfide, that rotten egg gas, dropped significantly below the human olfactory threshold, meaning even if a tiny microscopic trace was still in the air, a human physically couldn't smell it anymore.
Exactly.
Now, methylmercaptan did drop significantly, though it stubbornly stayed just slightly above the threshold. But the overall combined effect was a massive reduction in noticeable odor.
And beyond the gas, they also saw significant physical reductions in the subject's plaque index, plus a major drop in severe tongue coating.
Right, and we know that tongue coating is basically the prime real estate, the main breeding ground for these bacterial assembly lines.
Yes, so the physical environment of the mouth was demonstrably cleaner. But the microbiological data is where we really see the proof of that assembly line collapsing.
The PCR testing.
Right. The researchers used real-time PCR testing, which allows them to count the specific DNA copies of bacterial strains in the subject's saliva.
Yeah.
And the counts of Fusobacterium nucleatum, which is the gram-negative chef, often considered a bridge bacterium that anchors the biofilms, those numbers plummeted.
Wow.
The counts of Treponema denticola and Tannerella forsythia fell significantly too. And crucially, our gram-positive prep cook, Solobacterium morae, also saw a massive statistically significant reduction.
Okay, but hold on. I need to push back here because I was reading through these PCR charts and I noticed something that genuinely confused me.
Sure, what's that?
The data shows that the ClO2 mouthwash caused a massive drop in all those strains you just mentioned, but there were two major bacterial strains that it didn't seem to touch at all. Aggregatibacter, Actinomycetum comitans, and more importantly, Porphyromonas gingivalis.
Ah, yes.
Now, anybody who follows dental science knows that P. gingivalis is a major player in severe periodontal disease. And historically, it is heavily associated with producing odors.
Right, it's notorious.
So if this ultimate chemical assassin didn't actually reduce the P. gingivalis levels, doesn't that mean the mouthwash kind of failed at its job?
It is a completely logical assumption to make based on, decades of dental assumptions. But no, it absolutely didn't fail. In fact, this specific finding is arguably the most revealing part of the entire clinical trial.
Wait, really?
Yeah, it forces a massive paradigm shift in how we understand bad breath. Remember, the halitosis in these patients dropped drastically. The human organoleptic scores went down to basically 0.
Right.
The fact that the odor disappeared entirely, even while P. gingivalis levels remained perfectly stable, implies something profound about the oral microbiome.
Wait, so you're saying P. gingivalis isn't the main bad guy we thought it was?
Exactly. For years, dentists blamed P. gingivalis for almost everything going wrong in the gums and the breath. But this data strongly implies that P. gingivalis isn't the true linchpin of bad breath.
Oh, wow.
Instead, it proves that Fusobacterium nucleatum and our prep cook Solobacterium morae are the true foundational pillars of the halitosis cycle. Breaking their specific synergy is entirely sufficient to collapse the odor production, even if other notorious bad actors like P. gingivalis are still hanging around in the mouth. It is a brilliant revelation about how interconnected these bacteria really are.
That is wild. So what does this all mean for you, the listener, the next time you are standing in the pharmacy aisle feeling overwhelmed by all the minty marketing? Let's distill this journey.
Yeah, let's break it down.
We know now that bad breath is not just old food. It is a complex living ecosystem of gram-positive prep cooks and gram-negative chefs collaborating on your tongue to produce sulfur gases.
Right.
And based on this rigorous 2018 double-blind crossover study, the evidence strongly suggests that 0.1% chlorine dioxide is a highly effective, scientifically validated tool to stop them.
Absolutely.
It isn't just a flavor. It physically reduces the tongue coating where they live. It chemically starves the odor-causing bacteria by destroying their sulfur-rich amino acid food supply, and it chemically neutralizes the gases at their source.
And the practical takeaway here is that you need to fundamentally change how you look at the products in your bathroom cabinet.
Read the labels.
Yes. If you genuinely struggle with bad breath, you need to read the active ingredients on the back of the bottle. You want a compound that alters the oral environment like ClO2 does by introducing oxygen and penetrating those sticky biofilms.
Instead of just alcohol.
Right, you do not want a product that relies merely on masking the odor with a strong flavor or harsh alcohol. Alcohol-based washes often just dry out the mouth, reducing your natural saliva flow, which can actually make the environmental conditions much worse and encourage more bacterial overgrowth in the long run.
Exactly. You want the chemical assassin, not the cheap perfume.
Yep.
But as we wrap up this deep dive, I want to leave you with a final thought.
Okay.
Something to mull over that actually goes a step beyond what the researchers tested in this 2-week trial. We just spent all this time praising chlorine dioxide for its amazing targeted ability to dismantle this bacterial assembly line.
It did a great job.
It wiped out the prep cooks and the chefs beautifully over 14 days. But knowing what we now know about the incredibly delicate, complex balance of the human microbiome, if you use a powerful oxygen-flooding oxidant like ClO2 every single day for 5, 10, or 20 years.
Ah, I see where you're going.
Right. What happens to the long-term baseline ecosystem of your mouth? By essentially carpet bombing the bad actors with oxygen every single morning, could we accidentally be wiping out crucial beneficial bacteria that we might not even realize we desperately need yet?